Hello and welcome to some biology class. Friends, today we shall discuss about active transport. Active transport is a biological process by which ions and molecules are transported against their electrochemical gradient across a biological membrane. Because this process takes place against the electrochemical gradient, so it is dependent on energy input. In most of the cases, energy for active transport comes from the hydrolysis of ATP, but in few cases, energy also comes from light or from an already existing ion gradient. Active transport can be divided into primary active transport and secondary active transport. In primary active transport, energy comes directly from the hydrolysis of ATP. An example of primary active transport is Na plus K plus ATPase. This is also known as Na plus K plus pump or sodium pump. Na plus K plus ATPase is a p-type ATPase. It is an antiport because sodium and potassium ion are transported in opposite direction. This transport system was first characterized by James Coe. It is one of the most thoroughly studied active transport present in the plasma membrane of higher eukaryotes. Na plus K plus ATPase is a transmembrane protein composed of two types of subunits. Alpha subunit, which is about 1000 amino acids long, contain both the ATP binding site and the ion binding site. The other subunit is beta subunit, which is about 300 amino acids long. This subunit is not directly involved in the pumping of ions, but the beta subunit facilitates the correct insertion of alpha subunit into the plasma membrane. Therefore, alpha subunit is the catalytic subunit. In some cases, a smaller regulatory subunit known as gamma subunit is also associated with this protein. This figure explains the function of Na plus K plus ATPase pump. This is the protein and this is the lipid bilayer. This side is the cytoplasmic side and that side is the outer side. So the protein, that is Na plus K plus ATPase, drive the transport of 3 sodium ion out of and 2 potassium ion into the cell with the concomitant hydrolysis of ATP. For each molecule of ATP hydrolyzed, 3 sodium ions are pumped out and 2 potassium ion into the cell. It is because of this active transport system the sodium concentration is maintained at a low level and that of potassium ion at a high level inside the cell, whereas the situation is reverse in the extracellular fluid. Another type of active transport is secondary active transport. In secondary active transport, energy comes from an already existing ion gradient. So, in case of secondary active transport, an previously existing ion gradient is the source of energy. An example of the secondary active transport is the sodium glucose transporter, also known as Na glucose cotransporters or symporters, which is present in the apical membrane of the epithelial cells of the intestinal mucosa. This active transport system is also known as SGLT. So, the energy for this sodium glucose cotransport comes from a sodium gradient, and the Na plus gradient is established by the primary active transport system, that is Na plus K plus ATPase. This figure explains the process of secondary transport, which is carried out by sodium glucose cotransporter. This is the brush border cell, and these finger-like projections are the microvilli. This membrane is the apical membrane, and this membrane is the basolateral membrane. In the apical membrane, the sodium glucose cotransporter is present. With the help of this cotransport system, glucose molecules are taken into the intestinal epithelial cells along with sodium ion from the intestinal lumen. The energy for this cotransport comes from a sodium gradient. At the basolateral membrane, there is the primary active transport system, that is Na plus K plus ATPase. The system continuously pumps out sodium ion out of the brush border cells. So, sodium ion concentration is maintained at a low level inside the brush border cell, but outside the sodium concentration is higher. Due to the presence of this inward sodium gradient, when sodium ions are transported into the epithelial cell, along with sodium ion, glucose molecules are also transported into the brush border cell, from which glucose molecules can be transported into the blood circulation through a glucose uniport system, which is present in the basolateral membrane of the brush border cell. So friends, this is about primary and secondary active transport. In next videos, we shall discuss in detail about various active transport systems.